Environmental DNA is tiny fragments of genetic material that have been left in the environment by every living thing that passes through it. It includes things like fragments of skin, scales, slime, just about anything that has fallen off a living organism at some point. We can take samples from soil, water or air, process them in the lab and work out what the living organisms are that have been there. One thing that project developers might be saying about their projects is that they're looking after conserving rare species or species of conservation concern. So we can use eDNA to identify whether those species are present or absent from that project area. The hypothesis that we're testing with our work in Uganda is whether or not the type of bacteria that you find at a given site change in response to things like pollution events, other human impacts, agriculture and so on.
We collected composite soil samples and mixed them, and the aim is to analyze these samples and assess the species that might have existed in this area. And for the water samples, we collected them from all the streams that come in and out of the forest.
It's very much at pilot stage, but what we found backs the hypothesis up. We have seen different suites of bacterial species outside Kibale forest compared to the ones that we see inside. There also seems to be some kind of purifying effect of the forest. So the further you get into the forest, the more the bacteria that you see represent forest generally, rather than the influence from the outside. We are at the forefront of moving this science along in terms of the application of that technology to ratings on biodiversity claims. The ideal would be to have some kind of litmus test, I guess, in the future, whereby a project developer would be able to look for key indicator species, which would give some sense of whether things were improving within a project area compared to a reference outside of that area. We're still some way from that. This is early stages of research. We've now got new markets developing in nature credits, biodiversity credits, and so on. And those themselves will also need ratings in the fullness of time. Our hope in doing this work is that we can identify some of the basic properties of eDNA in this context and inspire other people to continue the research and develop those tools that we need.